Good day, Mom. Let me have an overview about the pentose phosphate pathway. The PPP is a multi-enzyme pathway that shares a common starting molecule with glycolysis or glucose-6-phosphate. The PPP gives rise to reveal critical products for cancer metabolism, including a pentose 5-carbon sugar or ribose 5-phosphate. Ribose 5-phosphate is a precursor for the synthesis of nucleotides and nucleic acids. Additionally, the non-oxidative phase of the PPP gives rise to erythrose 4-phosphate, which can contribute to the formation of aromatic amino acids such as phenylalanine, tryptophan, histidine, and tyrosine. PPP intermediates could also be converted to the glycerol or glyceraldehyde, rather glyceraldehyde 3-phosphate that returns into the glycolytic pathway, thereby creating a shunt from glucose 6-phosphate that bypasses the initial steps of glycolysis. Critically, converting one molecule of glucose to ribose 5-phosphate during the oxidative phase of the PPP converts to NADP plus 2 to NADPH. NADPH provides reducing power, electron, hydrogen donation ability for the synthesis of cellular building blocks such as lipids and cholesterol, and is also used to produce reduced glutathione, which controls reactive oxygen species or ROS. The enzymes of PPP are potential anti-cancer drug targets, as inhibition of the PPP would reduce nucleotide synthesis and increase ROS-induced cellular damage. As the first enzyme in the PPP or J6PD is the most common enzymatic deficiency in humans and has few ill effects, the PPP represents a cancer pathway that could be inhibited with limited side effects in normal tissues. So I have here an illustration of a summarize and its connections to other major pathways, which is the glycolysis, glycogen metabolism, and tricarboxylic acid or TCA. So, the various functions of the PPP can provide NADPH from NAD plus and ribose 5-phosphate for the ultimate synthesis of nucleic acids. NADH is needed to reduce GSSG to glutathione molecules joined by a disulfide bridge. The oxidized form of glutathione to GSH glutathione, particularly in cells such as the RBCs that are subject to oxidative stresses and the production of H2O2 and free peroxy radicals. The PPP is, in a sense, elastic in that it can adapt to the needs of a particular cell at a point in time when the metabolism of a cell is requiring reducing equivalents in the form of NADPH, or needing to divide that requires DNA and RNA, and the production by the PPP of ribose 5-phosphate, or needing to synthesize lipid from the same 3 carbon intermediates of glycosis, or needing energy in the form of ATP. I have here a 4 modes of pentose phosphate pathway. So, the 4 modes of the pentose phosphate pathway, each geared to the generation of a product needed by a particular cell. In mode 1, one molecule of J6P makes 5 molecules of 5R5P. In mode 2, one molecule of J6P makes 1 molecule of R5P and 2 NADPH. In mode 3, one J6P makes 12 NADPH, starting with 6 molecules of J6P oxidized to 6 ribulose 5-phosphates. The ribulose 5-phosphates can be rearranged by the pathway to form 5J6Bs. The overall stoichiometry is 6J APC plus 12 in ADPH by 5J6P plus 12 in ADPH plus 6CO2 plus P. In mode 4, 3 molecules of J6P make 6 NADPS, 8 ATPs, and 5 pyruvate. And NADH can be used to create more ATP. More ATP, 5 pyruvate, and that can be used to more ATP. So 5R5P or ribose 5-phosphate ATP adenosine triphosphate, while the 6G the J6P is the glucose 6-phosphate or NADPH nicotinamide adenine dinucleotide phosphate. Although the pentose phosphate pathway can completely convert glucose 6-phosphate into CO2, recycling the product fructose 6-phosphate to glucose 6-phosphate. The more usual products are glyceraldehyde triphosphate and fructose 6-phosphate that will then enter glycolysis. The pathway can therefore operate as a cycle depending upon cellular requirements. So, let's proceed to the citric acid cycle. So, the citric acid cycle utilizes mitochondrial enzymes for final oxidations of carbohydrates, proteins, and fats. Moreover, the Krebs cycle also produces intermediates which are important in gluconeogenesis, lipolysis, neurotransmitter synthesis, or etc. Acetyl-CoA from pyruvate of glycolysis, beta-oxidation of fatty acids, ketogenic amino acids, and ketones enter this pathway for energy production. The first step is fusion of the acetyl group of acetyl-CoA with oxalose state catalyzed by citrate synthase. CoAs and heat are released and the citrate is produced. Citrate is isomerized by dehydration and rehydration to isocitrate. The enzymes aconitase catalyzes these two steps using cis-aconitate as the intermediate. The next two steps are catalyzed by isocitrate dehydrogenase. Dehydrogenation of isocitrate forms oxaloxynate, which decarboxylates to alpha-ketoglutarate, or alpha-ketoglutarate is further oxidatively decarboxylated by alpha-ketoglutarate dehydrogenase, a multi-enzyme complex. Succinyl-CoA is formed in this unidirectional reaction. Thank you, ma'am. That's all.